cytochrome C oxidase, which is now considered the primary molecule which absorbs light and which is responsible for those reactions what we see in cellular work and in clinics. And this chemical part and this photoacceptor, they are only a little part here, this so-called redox-sensitive part of cytochrome C oxidase, and this is a huge globe of protein around it. And a simplified system, a simplified scheme shows how electrons arrive from cytochrome C, here, this part. And first, there are two atoms of copper, so-called copper A, as I told. Then there is porphyrin, heme A and heme A3. And this part, together with copper B, it is so-called reactive center of enzyme. This is a part where the chemical reactions with oxygen are occurring. So during years we have proposed at least 4 possible primary reactions, and first we proposed first possibilities when it was not clear which is the primary photoacceptor molecule. And today I would like to underline 2 of those possibilities. This one was really the first one. I proposed in '88 changes in redox properties and acceleration of electron transfer. And that time we didn't know exactly in which molecule it occurs. And another possibility, so-called NO hypothesis, proposed in year 2000. Both of those possibilities are connected with redox reactions. So, about redox properties alteration hypothesis, there was only one possibility how to check this, and we had to build up apparatus and spent many years to study, to write absorption spectra of cellular monolayers, which is, from spectroscopical point of view, a very non-easy task. Then this apparatus was made together with Institute of Spectroscopy in Troitsk, and 2 things we wanted to make, to compare the action and absorption spectra in this important region for laser therapy, and to see if there are any changes in absorption spectra due to the irradiation. It was again big work, and we studied it, and we didn't know how to quantify our results. And here I would show you 3 examples. This spectrum, you see, this is a very low absorption. This is in an enclosed, when the cuvette of measurements is closed. It is open here, plenty of oxygen here, and this is dry monolayer. There are no chemical reactions anymore. And in this way, playing in those spectra, we found out that it is possible to quantify those spectra by intensity ratios of 2 peaks, 760, 665. And in this way, we were able to characterize which state is our photoacceptor, in more oxidized, relatively more oxidized, or relatively more reduced. you will see changes after irradiation by 8:30. And of course, again, you will see that comparing those parameters, and here you see there are differences. And I will not explain this again, my time is short. But when you compare those absorption spectra with action spectra about DNA synthesis rate, reactions occurring in one part of cells, and those action spectra about radiation in another part of cells, you see that there is a real similarity, and you can also characterize absorption spectra by this particular way and compare it with absorption spectra. So we have found out that photons really have a direct stimulatory effect on cytochrome C oxidase, and the effect is that electron flow between heme A and copper B, A3 reaction center of cytochrome C oxidase, it is increasing, and it causes transient reduction of the enzyme, and which is followed by relative oxidation. It means that by irradiation we perform a transient upgrading or activation of cytochrome C oxidase. And this is another example of those complicated experimental results. I will not stop from them because I would like to show you the following thing. For example, when irradiating with helium neon laser, you two sets of experiments. Initial spectrum is relatively oxidized and initial spectrum is relatively reduced, and you see when you build up some dose dependencies, you see the nice curves. This one, everybody has seen this, I think. This increasing part of curve is maximum, decreasing part. But if you work a relatively reduced enzyme, there are many electrons already in center, you get the opposite effect. And at least in our cellular effects, we have sometimes got such kind of dose dependencies as well. And we didn't understand why sometimes it gets in this way, but not in this. And you see the maximum is in the same place. And there are really certain borders where you can use light to increase this reaction. It is not coming for forever or like something like this. It is very certain borders, and it means that really we have seen, everybody of us, in tissue work, for example, or so in cellular work, when cells are or tissue in nice conditions, you really cannot do by irradiation something. It works nice only in damaged cells. So concluding this part, we can say that irradiation causes reduction or oxidation effect in cytochrome C oxidase. It depends on initial redox state of the enzyme, and there are certain values, borders of irradiation effect. In our case, it is between there. So let's see now this possibility. We spoke about this. We understood many things. Let's see what about this. It is, I think, known maybe 10 years or maybe less that NO is a psychological inhibitor of cytochrome C oxidase. It is present in real cells, in real organisms, in very low concentrations, and in this way it can change, or it modulates the effect of working of cytochrome C oxidase. And in other ways, it is connected with breathing. And it is very well known that it is bind to cytochrome C oxidase reaction center. This is the reaction center of cytochrome C oxidase. This is the heme part, and this is the copper part here. And NO can bind from this side or from this side, and also from both sides, and it can be removed very easily. And light is one component which can regulate this reaction. And this is not so important. I wanted to bring some examples, some comparing with one other chemical, azide, which is also binding to cytochrome C oxidase center, but it is there forever. It will not be removed by irradiation, for example. And so there is one set of experiments. We measured cell adhesion and NO, or this azide were present, and these action spectra were different. And not again giving details about all these things, we showed that in NO hypothesis is a working hypothesis. It can happen here, and light really can remove NO for some time from reaction center, and in this way the reactive activity of enzyme is increasing. It means that in irradiated cells, respiratory chain, it acts as signal generator and also as signal transducer. And this is maybe even the more important feature of this reaction, that after relieving some blockade of enzyme, for example, blockade caused by NO, and in this case we get a reverse of signaling consequences, and this is really important thing, how the respiratory chain is ruling the metabolism of whole cell. Now I spoke about mitochondria here, but we started from this part of cell, from nucleus, and how those signal from mitochondria is given to cellular nucleus. This is an important question arising. And here I wanted just to remind you that this very nice experiment performed some years ago. They used DNA microarray analysis and gene expression. Almost 10,000 genes activity was changed after irradiation with 628 nanometers. And among those genes, you see this one DNA synthesis which we measured. I started with those spectra about proliferation and many other things also. This is a very remarkable work, I should say. Yes, this is one more time to underline why this part of cellular mechanisms after absorption of light is so important, and how the spatial and temporal separation of different reactions can be explained. But now, let's